You know, some of the most essential products in our world, things we rely on every single day, are made possible by a hidden microscopic workforce. We're going to dive into a technology that powers pretty much everything, from our medicine to our food, but not with silicon and steel. No, this is about harnessing the fundamental processes of life itself.
And it all starts with stuff you see every day. That bread in your kitchen, the cheese on your plate, these are actually ancient examples of fermentation. It's a process we've now scaled up to this massive industrial level, turning really simple ingredients into complex flavors and nutrients.
But it goes way, way beyond the kitchen. This exact same core technology is absolutely critical for modern healthcare. I mean, think about insulin. Before this, diabetics had to rely on animal sources. But today, we use engineered microbes to produce pure human insulin, saving millions of lives. And the same goes for so many vaccines and other complex drugs.
And it's even helping to power a more sustainable future. Biofuels, like the ethanol that comes from corn, are a direct result of putting these tiny biological factories to work for us, turning renewable biomass into clean liquid fuel. So here's the crucial point. All these incredibly diverse products, they're all connected. They're all made through a process called large-scale fermentation. And that process is this amazing bridge that takes us from a single invisible cell all the way to vast global supply chains.
So what is it exactly? Well, large-scale fermentation is a super precisely controlled process where we use microorganisms, think bacteria, yeast, fungi, to convert raw materials like sugars into really valuable products. It's the absolute heart of industrial biotechnology. It lets us build these complex, three-dimensional molecules that would be incredibly difficult, if not totally impossible, for traditional chemistry to make efficiently.
All right, so now that we know what it is, let's take a look at how it all works. We're going to head inside the industrial engine, which really just comes down to two essential components, the cell and the vessel it lives in.
So first, the vessel. This is the fermenter, sometimes called a bioreactor. But don't just picture a giant steel tank. No, this thing is a highly engineered, perfectly sterile environment. You can think of it as the chassis that holds and protects our powerful biological engine.
And if we take a look inside, you can see just how sophisticated this chassis really is. You've got the agitator, right? Which is basically a powerful mixer that makes sure every single cell gets the nutrients it needs. Down at the bottom, there's a sparger bubbling in oxygen, and that outer shell, that's a cooling jacket. And it's vital for getting rid of the immense heat that these microbes generate while they're working.
Okay, we've seen the hardware, so now let's meet the living software. It's time to zoom all the way down to the microscopic level and get to know the organisms that are doing all the heavy lifting.
First up, we've got bacteria. These guys are the sprinters of the microbial world. They grow super fast and have pretty simple needs, which makes them perfect for producing things like proteins and amino acids quickly and efficiently.
Next up, yeast. These are your robust workhorses. They're incredibly tough and can tolerate harsh conditions, like low pH or high alcohol concentrations. That makes them the classic choice for producing ethanol, all sorts of beverages, and even some really complex pharmaceuticals.
And finally, we have fungi. You can think of these as the master secretors. Their unique structure lets them produce and then pump out huge quantities of complex products, like industrial enzymes or, the world-changing antibiotic penicillin, which completely revolutionized medicine.
So this table really lays it all out, doesn't it? It shows how we choose the right microbe for the right job. Bacteria bring the speed, yeast brings the toughness, and fungi, well, they're the experts at secreting complex molecules. Picking the right organism is the first, and maybe the most critical, decision you have to make.
But here's the thing, just having the right microbe isn't enough, not by a long shot. To get them to perform at their absolute peak, engineers have to create the perfect environment. I'm talking a 5-star, all-inclusive resort that's tailor-made to make them as productive as humanly possible.
And that process has 4 crucial steps. First, you prepare the perfect nutrient broth, or media. Then comes sterilization, and this is a non-negotiable step, using high-pressure steam to kill any contaminants. Next, you grow up a big, healthy starter culture. And finally, the fermentation itself, where every single parameter, pH, temperature, oxygen, is continuously monitored and controlled with incredible precision.
Now, industry uses a few different strategies to run these things. The simplest one is called batch mode. It's just like it sounds. You put all the nutrients in at the very beginning, close the lid, and just let it run until all the food is gone. It's simple, but it's not always the most efficient.
So, building on that idea, the most common industrial method is called fed batch. Instead of dumping everything in at once, you feed the nutrients in over time. This keeps the cells from getting stressed out and lets them grow to much higher densities, which at the end of the day means you get a lot more product. And then, for maximum efficiency, you have continuous fermentation. In this setup, fresh media is constantly being added while the product is being removed at the same time. This keeps the microbes in this permanent state of high productivity. It's incredibly powerful, but as you can imagine, it's also the most complex and difficult to manage.
Okay, now this all sounds great in theory, right? But let's talk about the immense engineering puzzle of taking a process that works perfectly in a little lab flask and making it work in a vessel the size of a small building. This, right here, is the great challenge of scale.
And with all of this complex machinery and biology, you might think the biggest problem is some kind of mechanical failure, like a pump breaking. But that's not it. In reality, there is one single risk that towers above all the others.
Contamination. That's it. A single foreign microbe getting into that pristine system can outcompete your workforce, spoil the product, and completely destroy weeks of work. I mean, a single breach in sterility can wipe out an entire multi-million-dollar batch. It is the one thing that keeps plant managers up at night. Absolute sterility is rule number one, period.
And, of course, there are other major hurdles. You get a lot of foaming, which can clog up filters and, you guessed it, create a pathway for contamination. It also gets exponentially harder to deliver enough oxygen and remove enough heat as the tanks get bigger. And of course, the cost to build and operate these massive sterile facilities is just enormous. So, where does this all lead? Well, this incredible technology is not just standing still. It's a dynamic platform for innovation, and we are now entering an era where the future is truly being built with engineered biology.
You see, with every advance in synthetic biology, we are upgrading this biological engine. We're getting the power to design life with more and more precision. And that leaves us with a really fascinating question. As our mastery of this process grows, what are we going to do with it? Are we going to engineer microbes to create self-healing concrete? Or maybe plastics that biodegrade on command? Or even food that's grown from thin air? What monumental global challenge will we solve next?